All right, I'm going to go through this last page in the notes. This is optional. This will help you answer a level 4 question that will be on your test. And before I talk about, though, some additional details about the chloroplast and mitochondria that kind of have left researchers scratching their heads, we need to review again bacteria cells and some of the features in them. Remember, bacteria cells are different than eukaryotic cells. They have one DNA molecule that's circular, and they do have one organelle, and that would be ribosomes, because DNA codes for proteins, and the ribosomes would then have to build the proteins for the cell, okay? So after reviewing those features, let's talk about what researchers have discovered over the years about mitochondria and chloroplast. When we look at chloroplast, we see something interesting about them. Is one, they actually have their own DNA. And we just learned that cells have DNA, right? So why do these organelles have their own DNA? And we also see their own DNA in mitochondria as well. Something else we discovered is that they have ribosomes. Again, this is an organelle. So why does an organelle, or how is it that this organelle has its own ribosomes, can make its own proteins? Same with the mitochondria. The mitochondria has its own ribosomes as well, okay? Something else that it kind of sets it apart from other cell parts is it has two membranes. The mitochondria does. When we take a look at the chloroplast, we see it has two phospholipid bilayers around it. It has two membranes. All the other organelles inside cells have one, like the smoothie R, the Golgi, and so on, okay? So when we discover something in science, we have to come up with a logical explanation that has to be supported by what it is that we know is true about, in this case, the mitochondria and the chloroplast. So what could explain why they have these features, okay? So I'm going to go down here to this little part right here. And let's see if we can devise a logical explanation as to how we ended up with cells that have mitochondria and chloroplast in them, but yet themselves, the mitochondria and chloroplast, have their own DNA and their own ribosomes, and so on. So let's draw a little bacteria cell. So here's a bacteria cell. And let's put its own circular DNA, because we know bacteria cells have circular DNA, and they also have ribosomes. I've shown you many videos in this unit, and I've shown you videos where a bigger cell could wrap around a smaller cell and bring it in and have it now inside in a membrane-wrapped vesicle. So here's that original cell now with its DNA and its ribosomes, just now living inside of a bigger cell, okay? Now, that would explain how we can have structures with their own DNA and their own ribosomes, and now they have this structure. If you look here, it's got two membranes around it. So its original membrane that it had, because it's a cell, and then this vesicle is a membrane that comes from this host cell. So it would have two membranes around it. And so the idea that attempts to explain why mitochondria and chloroplast have these unique structures is called the endosymbiotic theory. And a theory is an idea of science that is backed by an incredibly large amount of evidence. And we haven't found anything to contradict this idea. And so this is what the endosymbiotic theory states, that billions of years ago, when we had very, very simple cells, some cells could do simple processes like make ATP. Some cells can make simple processes, are photosynthetic, and they can make glucose. What if they ended up getting inside of bigger cells and started living inside of the bigger cells? And these bigger but simple cells still, because this was billions of years ago, we didn't have really complex cells at the beginning, as you know, when cells were evolving. They just didn't have the ability to digest and break down these other structures that got inside. So they just started living in there. So another idea that, or observation that would support that mitochondria and chloroplast evolved from single prokaryotic cells that were engulfed by other cells and then started living within those cells, is that if you look at chloroplast and mitochondria, they divide just like bacteria do. They'll elongate, they copy their DNA, and they split in two. So that's how all chloroplast and mitochondria come to be about, is the cell itself doesn't make it, the chloroplasts make themselves. They divide into two. The mitochondria divide into two. So if this cell, now living inside of a bigger cell, divided, we'd get several mitochondria or several bacteria cells that could make ATP. And then if this divided, you get several photosynthetic bacteria that can make glucose. And then if this cell then divides in half, because we know cells can divide into half, then we would have two cells that would have these bacteria living in them, some making glucose and some making chloroplast. But that would explain how cells, even today, ended up with mitochondria and chloroplast, because this happened billions and billions of years ago, and these cells are the ancestors of living things that we have today. And these features have continued to be passed on from generation to generation, okay? So you should be able to tell me what are some observations or evidence or features of mitochondria and chloroplast that make us believe this is how they came to be about inside of eukaryotic cells that we see today. They have their own DNA, it's circular. It even has matching genes on it compared to genes that we find on bacteria today. They have their own ribosomes. Those ribosomes actually look really similar to bacteria cell ribosomes. They divide in a similar way as bacteria. They're about the same size as bacteria, and they have two membranes instead of one, because there's no other cell parts in the cell that have these features. The rough ER, the Golgi, we have a completely different idea of how those came to be about structures inside of the cells, but different from the mitochondria and chloroplast.